Alright, biologists, this is membranes and membrane transport, theme B, form and function, topic B2, cells, subtopic B2.1. So every cell has a cell membrane. It's fundamental. Every cell has a cell membrane. Not every cell has a nucleus, but every cell has a cell membrane. And the cell membranes are what we call lipid bilayers. You see, they are bilayers because one membrane is made of two layers, one here in pink that we see and one in green. And they're fairly symmetrical, one kind of upside down in relation to the other. And they are made of this structure here, which is what we call a phospholipid. Now, observe that this is not two membranes. This is one membrane, and one membrane has two layers. So this whole thing here is just one membrane. But why do membranes have two layers? Well, to answer this question, we have to take a closer look at the phospholipid. You see, phospholipids have this part here, which is a hydrophilic head. This hydrophilic head is polar. It has a phosphate, which is an ion, which has an electric charge. Therefore, it's very polar. And polar things like water, if you remember from theme A, water is a very polar solvent. And because it's a very polar solvent, it's really good in dissolving things that are also polar. But if the phospholipid was entirely hydrophilic, entirely polar, then the membranes would dissolve in water, and that's not good. We need the membrane to separate the outside from the inside of the cell, and both sides are aqueous solutions. So the membrane cannot dissolve in water. That's why it has these two hydrophobic tails. And these hydrophobic tails are made of fatty acids. If you don't remember what fatty acids are, you should take a look here at the carbohydrates and lipids video. But the point is, since the hydrophilic head loves water and the hydrophobic tails don't like water, the membrane spontaneously arranges in this shape with the hydrophilic heads pointing to the water, both out of the cell and inside the cell, and the hydrophilic tails in the middle, away from water and in contact with each other, holding the membrane together. So because these hydrocarbon tails, these fatty acid tails, are hydrophobic, they do not allow the diffusion of polar molecules or hydrophilic molecules, or even large molecules. Therefore, the lipid bilayer acts as a barrier, which is great. Imagine you are a glucose molecule, very polar, very big, and you're inside a cell. Well, you don't want your glucose molecules leaking out of the cell. Because of these hydrocarbon tails, these hydrophobic tails, glucose cannot diffuse out of the cell, and the cell membrane acts as a barrier, preventing amino acids and glucose and organelles from just swimming away out of your cell. Now, that does not mean that nothing can go in and out of the cell. There's a thing called simple diffusion. If you have a difference in concentration from outside to inside of the cell, for example, here we have this small molecule, this small red molecule, and we see that the concentration out of the cell is much greater than the concentration inside the cell. Diffusion is the movement of these particles across this membrane. Actually, for diffusion to happen, you don't need a membrane. But in this case, you have different concentrations on different sides of the membrane, and the particles will simply diffuse, will simply move from one side to the other until the concentrations are balanced. This can only happen to nonpolar or small molecules like carbon dioxide and oxygen. What if I want to transport larger molecules or polar molecules or ions? Well, then you're going to need proteins. And there are two types of proteins in the membrane. Integral proteins are proteins that are embedded in the membrane. So this protein is integral. This one, integral. This one, also integral. This one, integral. What about this one? it doesn't go all the way through, but it's still embedded in the membrane, therefore it's integral. What about the peripheral proteins? This is a peripheral protein. Peripheral proteins only touch the they only touch the surface of the membrane. They're mostly inside the cell, otherwise they would randomly diffuse away. So mostly peripheral proteins are inside the cell and they will only touch the surface, right? When you're drawing up, if the question asks you to draw a diagram, your peripheral protein cannot enter the membrane. It can only be on the surface. So these proteins can have all sorts of functions. They could be receptors for a hormone, for example. It could have a hormone or a messenger or a neurotransmitter. And then you have a receptor that attaches to the protein in the membrane. Or this protein can act as anchorage for the cytoskeleton or even extracellular matrix proteins. This protein could act as signaling protein, or it could even be a transport protein with a channel here. So the movement of molecules across the membrane is called diffusion. But if we are talking about the movement of water, this is osmosis. Even though water is polar, it's still very tiny. The molecules are very tiny and they can pass between the phospholipids. So we say that the membranes are semi-permeable, meaning they can let some things through. Not everything, but some things. They're permeable to water, a few solutes like oxygen and carbon dioxide, but they're not permeable to most things. The net movement of water, that means taking movement to one side and the other side, taking everything into account, the net movement of water molecules across a membrane, in this case you have to have a membrane, due to difference in water potential is called osmosis. What does that mean? Look here at this image. You have a high concentration of solute on one side and low concentration of solute on the other side, which means here we have proportionally more water. It's like the concentration of water is higher, but we don't say concentration of water because water is not a solute, it's a solvent. So we say that this has a higher water potential. So from a region of high water potential to a region of low water potential, you have this movement of water because the membrane does not let the particles move, but it lets water move. So how do we keep the concentrations the same? Instead of moving the particles across the concentration gradient of the particle, we go the opposite way. We bring water to kind of dissolve, to dilute the other side. And this movement of water to dilute the other side, this is called osmosis. Now, some cells have special proteins that can accelerate this process called aquaporin. Aquaporins are transport proteins. They're like channels, but for water. You find those proteins in cells that are specialized in water transport, such as in the kidney that has to reabsorb loads of water back in the blood, or in root hair cells in plants that have to absorb a lot of water from the soil into the plant. So you see, this is the lipid bilayer here, and we have this protein. You can see the protein as being sliced here in half. So we're seeing the sliced protein, and we see that there's a channel here in the middle that is just big enough to let water pass through. And we see that the middle of this channel is polar because water is polar. So this is a channel that is specific to transport water. Right, so imagine you have loads of this polar molecule here, this molecule that cannot diffuse through the membrane. So instead of this hexagon coming down here, since this movement is not allowed, now cannot do that because the membrane acts as a barrier. So what can we do? We can transport water to the other side, and now we can balance the concentrations. What if I want the diffusion of an ion to happen? What if I want diffusion of glucose to happen? I want glucose to enter the cell, or I want sodium or potassium to cross the membrane. and they are polar. How can I do that? You can have channel proteins that are specific for transporting this specific ion or molecule goes through this specific channel. Channels are not general. Channels are specific for each solute. So we have different channels for sodium, for potassium, for calcium, chloride, so that we can control better what goes in and out of our cells. But the problem with diffusion, simple or facilitated, is that it only happens as long as there's a difference in concentration. It only goes from the more concentrated to the less concentrated. What if you want transport to happen beyond the equilibrium? What if you don't want to reach an equilibrium? What if you want to go from the less concentrated side to the more concentrated side? You want to go against the concentration gradient. Then you need active transport. It's called active because it requires ATP. It will spend energy. And for that, you require a protein. This kind of protein is also known as pump because they will pump your solute from one side to another. They will push against the concentration gradient using energy from ATP. So to recap, you can have simple diffusion when the molecule can pass through the membrane without assistance from the most concentrated side to the less concentrated side. You can have facilitated diffusion, which is just like diffusion, but you need a door, you need a protein to act as a channel. And then you have active transport. Active transport will push from the less concentrated side to the more concentrated side. It's going to use a pump protein and it will spend ATP. And as we mentioned before, facilitated diffusion is selective. You have specific channels for specific solutes, for specific molecules or ions you are transporting. Simple diffusion, on the other hand, it's not selective. It only depends on the concentration gradient. And because there's no protein involved, that's the only thing that will drive the movement. As long as the molecule is small enough, non-polar, it can just go through the membrane based on the concentrations on one side and the other side of the membrane. But facilitated diffusion, as we said, again, is selective. The reason is the channel has to be the right size and the right polarity to allow the transport, right? You see here the selectivity filter, meaning if the molecule is too big or it has the wrong charge, it will not pass through. Now, glycoproteins and glycolipids, as the name might suggest, glycoproteins are proteins conjugated with a carbohydrate. So it's a protein that has a carbohydrate attached to it. Now, glycolipid, it's the same thing, but it's a lipid with a carbohydrate. And why do we have that? Glycoproteins are great for cell recognition. They act as badges that say, hello, my name is this kind of cell, because cells don't have eyes. How do they communicate with each other? For example, through glycoproteins. Also, glycoproteins are also involved in cell adhesion to keep the cell in place and maintain the stability of the tissue. Glycolipids, not super common, but they are important because they contribute to the stability of the membrane by forming hydrogen bonds with the water. Remember that carbohydrates have loads of OH groups, and these OH groups form hydrogen bond with water, and the hydrophobic tails or the hydrophobic tails of glycolipids will anchor that to the rest of the membrane. So here we see the different blood types, type A, B, AB, or type O, depend on these carbohydrates that we have on glycoproteins. And finally, the fluid mosaic model of membrane structure is the name of the model that we just learned. It's called a fluid mosaic because the parts are not stuck in place. The proteins are free to float and diffuse throughout the entire membrane. They're not fixed in place. They're spinning and moving, going from side to side. Everything is fluid. The lipids are fluid, and all these parts act together as a mosaic. It is to contrast the previous model of the sandwich model. It was already dismissed But it made sense if you look at the electron micrograph of a cell membrane. This is a cell membrane, and it had a darker layer, and a lighter layer in the middle. Let me change colors here. So we had a darker layer, and a lighter layer in the middle, kind of forming a sandwich. So people used to think membranes had proteins on the outside and lipids on the inside, forming a sandwich, but this model has been disproven by many experiments. Now we know that the proteins are embedded, mostly the integral proteins are embedded in the membrane, forming a fluid mosaic.